The webinar will be steered by two leading doctors, both well-known names in the research and medical field, who have made our community proud in many ways. It is with great pleasure that I introduce you today to these two wonderful doctors. Dr. Villoo Morawala-Patell, the founder and chairperson of Avesthagen Limited in Bangalore, and Professor Dr. Keki Turel, Emeritus Chairman, Neurosurgery, Bombay Hospital. Let me start with a brief introduction on Dr. Villoo Morawala-Patell. Dr. Villoo Morawala-Patell, founder and CMD of Avesthagen Limited and the Avesthagenome Project, is an innovator and has been involved in international biotechnology development, academic and corporate leadership, and forged alliances with academia and industry. She is a recipient of many national and international awards as a scientist and as an entrepreneur, including the French National Order of Merit, Karmavir Puraskar, Parsi Entrepreneur Par Excellence Award, and Outstanding Women Entrepreneur of the Year Award. Dr. Patel is also a recipient of J.N. Tata and Rockefeller Foundation Fellowship. Her international biotechnology experience, coupled with her long-standing interest in global development, led her to founding Avesthagen Limited, a company built around the convergence of pharma, food, environment and just agriculture, and population genomics, leading to predictive, preventive, and personalized healthcare. Speaking about Professor Dr. Keki Turel, he is our community's leading medical luminary. He commands 47 years of expertise in neurosurgery and is a consultant neurosurgeon, Professor Emeritus, Department of Neurosurgery at the Bombay Hospital, Institute of Medical Sciences. Dr. Turel is the Managing Trustee at the Mumbai Institute of Neurosciences and the founder chairperson of International Conference on Complications in Neurosurgery since 2017. He has held leading positions in several prestigious and medical associations, including the Neurological Society of India, Associations of Surgeons of India, and more. A pioneer in microsurgery, he has performed extraordinary medical procedures and removed the world's largest brain tumor, weighing 550 grams, at the Masina Hospital and another of the same size at the Bombay Hospital. His other marathon sessions include non-stop operations for over 26 to 27 hours during removal of brain and spinal cord tumors. He founded the neurosurgery department in Oman in 1980. He was the first surgeon in India to operate on an AIDS patient in 1989. Known for his compassionate outlook, he has offered his time and expertise to treat the less fortunate. He is a proud recipient of over 18 national and international awards. He has delivered over 700 lectures in 67 countries. At a personal level, he is an ordained priest of Zoroastrian faith and a professional photographer and filmmaker. Okay. All right, Dr. Turel and Dr. Villoo Morawala-Patell, will you please start your session?
Sure. So let me first begin by thanking WZCC and Meher Besania for inviting me to be moderator of the first of the series of webinars they intend to have on development of the Zoroastrian community by partnering with businessmen and professionals. It's my distinct honor and pleasure to share the stage with Dr. Villoo Morawala-Patell, who has already been introduced, albeit briefly due to want of time, but one could speak volumes about her researches, scientific zeal and achievements, and of course the entrepreneurship in the booming field of biotechnology through the formation of Avesthagen Limited. Dr. Villoo, you have been closely following the history of Parsis and presently unraveling the past of ancient Zoroastrian-Parsi genome and its implications, not only for our own future, but even for the community of the world. Before we proceed to speak about Dr. Villoo, and since most of our viewers may not be medical people, let me simplify the few scientific terminologies we would be encountering during this conversation. The human body is made up of cells, 37 trillion cells of 50 different subtypes. Cells, as you know, are fundamental structural and functional unit of a living organism. It is likened to a raw egg, if you can imagine the raw egg having the yellow nucleus and the egg white representing cytoplasm. This yellow nucleus contains 23 pairs of chromosomes. That means 46 chromosomes. And each chromosome contains DNA, which is in the form of a double helix, and that transmits genetic material. The genes are a distinct sequence of DNA, and they are the biologic unit of heredity. Cytoplasm, which I liken to the egg white, contains rod-like structures we call mitochondria, and they provide energy to the cell and contains its own DNA too. Genome, what Dr. Villoo is going to talk about, is a complete set of genetic material along with the hereditary factors. And she will now talk about her research on the Zoroastrian-Parsi genome or the ZP genome. So Villoo, are you ready to take my first question?
Sure.
Okay. So my first question is that you have, through your Avesthagen Limited, recently released the first complete Zoroastrian-Parsi mitochondria reference genome. I would like you, for our listeners, please explain what is this study about. The next question, of course, is what is the mitochondrial genome, as the title suggests? What are its implications compared to the nuclear genome? And what, according to you, is the most promising area of your research? I hope there are not too many questions.
Quite a load full, but I'll attempt. I'll start with the history first of this group of people whose ancient homeland, as mentioned in the sacred Avestan text, Vendidad, and was near the North Polar Arctic region. And as has been also certified by B.G. Tilak in his publication, was the home of the Vedas, and has been corroborated by Bennett that the Indo-European culture originated in the Hyperborean regions of northern Siberia and the islands of the circumpolar regions. Now, we are talking 12,000 years ago. This region suffered a natural calamity and became ice-clad, necessitating the southward migration of its pastoralist inhabitants. And by 4000 BC, the Indo-Europeans took over the Eurasian steppes. From that late second to the first millennium BC, the Indo-Europeans, mostly on the basis of religious worship, split into Indo-Aryans who moved further south and crossed the Hindu Kush, while the Indo-Iranians, the Medes, the Persians, the Parthians began populating the western portion of the Iranian plateau, close to the Alborz and the Zagros mountains, and northern Mesopotamia to southeast Anatolia, what is now called the Fertile Crescent, where significant innovation in agriculture occurred. The Zoroastrian-Parsis today trace their origin to these pastoralist migrations to the Eurasian steppes. They populated this western Iranian plateau, and the settlement flourished and established what were then the Zoroastrian Persian empires. During the time of those Persian empires, the Achaemenid and the Sasanian empires, Persia became a global hub for culture, religion, science, art, gender equality, and technology. So this is the travelogue of that mitochondrion that we have today with us. And post the defeat of the Persians by the Arabs in two decisive battles in 636 and 642 AD resulted in the fall of the Zoroastrian Persian Empire. It was only in the 742, 100 years later, that a boatload of people from the district of Pars and those Khorasan regions arrived in India and they escaped persecution and they arrived in India. And these are the people we are talking about. Their history is ancient, and their mitochondria, as we discovered from our work that we did, their mitochondria also are ancient. So that's why I call it the travelogue. It's been a journey of migrations and of communities that survived and built a new life in India. So just to tell you what you asked, what's a mitochondria? Human mitochondrial DNA is a circular double-stranded genome, just 16.6 kilobases of bacterial origin. Actually, mitochondria, the cell, the initial early stages of cell engulfed a bacteria to give it its power. So the mitochondria is a powerhouse and is a vital and encodes genes, subunits of the energy generating electron transport chain pathway. The analysis of the variability of these and the mitochondria's most significant trait is it's maternally inherited. It comes from the mother, and the egg carries the mitochondria, while the sperm also carries the nuclear material. The egg carries both nuclear and the mitochondrial information. And the mitochondria, the take home is encodes for the genes, for ATP production, for respiration, for energy. And so many of those genes are extremely important for our lives and our longevity, as well as many rare diseases and interesting outcomes. So, and it has been used to reconstruct the history of populations with respect to maternal inheritance. It's in many cases across the world, they have used the mitochondria to describe inheritance, to track back migration histories, to track back even all the way right down to the first mother in Africa. The accumulation of these the mitochondrial variants, when I say variants, these are differences. You know that most of us are 99.9% similar, but there are certain differences which are referred to as changes in the DNA code, referred to as variants or single nuclear polymorphisms, or there are many such labels. The accumulation of these mitochondrial variants through maternal inheritance has a role in tracing haplotypes. What are haplotypes? Haplotypes are subgroupings of humanity. And you may say it's like the caste system of India. There's a general overarching caste, then there is the subcaste, then there's the sub-subcaste. So those definitions can be, have not yet been linked to haplotypes today, but these conservation of certain variants in certain subtypes result in a fallout of what are known as groups called haplotypes. And so and is used to follow these genealogies, maternal genealogies of history and migration. And such an approach gives insightful findings into the origin and disease etiologies associated with endogamous communities, as was proven by the Icelandic population study in 2005, 6, 7. So the most promising work of our current work in terms of innovation is the significant and prolific outcomes we are seeing from the Avesthagenome Project, largely because of the endogamous nature of the community and the ability to track those genes. They are not searching for a needle in a haystack, but very, very conserved, easy to find for cancers, for neurodegenerative conditions and rare diseases. And our recent work on the genomics of the Zoroastrian-Parsis converges the fallout of ancient history, human migration, endogamous population genetics, social behavior and customs that express in our community in genetic signatures of contributing towards wellness and health, ultimately as the outcome from this study. our project started in 2007, 2008, when I realized that the community was fast dwindling. And I found myself on that cusp where, I knew what was needed to be done coming from that genomics field, and being a Parsi also added to that. And I said, this is the time and moment when we have to capture and encapsulate this biodiverse, archive this community so that we can continue to contribute in the future. So this became sort of a role and work I had to shoulder, and elucidate the journey of this community and the impact of their genetic makeup. We collected in total 4,600 blood samples from almost 9 cities in India, from Bangalore, Hyderabad, Surat, Navsari, Bombay, Chennai, Delhi, Calcutta. And it was a fantastic experience. We went around from these cities with a bunch of scientists and clinicians. And so the collection was in a manner that it took care of all the new omics technologies. So from genomics to stem cells. So we collected 9 tubes. So it's a very thorough archiving of our blood samples. So you can elucidate not only at the genetic level, but at the exosome level, like when you express, as well as at the protein level, and then ultimately the stem cells. And we also collected 65 pages of data, the questionnaire purpose, which was very well put together, and that provided information on the person, individual as such, as well as his family backgrounds, his food and medical habits, and all of it was captured. So in this study, so this is what we call the archive. We have this archive today of 4,600 Parsi samples, as well as the information, and it's a very big asset which can be used for years to come. Phylogenetically and statistically, this publication focuses on the analysis of 100 Parsi mitochondrial genome sequences, which we sequence. Mind you, this is the human nuclear genome. The nuclear genomes have also been sequenced, and the analysis is going on. The mitochondrial sequence is the extracellular, chromosomal material, that extranuclear chromosomal material we discussed. And so it's focusing, so the data we are today talking about is focused on 100 Parsi mitochondrial genome sequences collected in Hyderabad and Bangalore, as Hyderabad, Sikandrabad. And it's also the year of our celebration of the Parsi Agyari, our Chennai Agyari in Sikandrabad. And I wish them all the best. So we collected the samples there. And so this is a very clear study that was done. And disease association mapping showed that the majority of the mitochondrial variants were linked to longevity and its associated conditions, revealing the genetic basis of many of the heritable diseases in the community, like cancer, neurodegenerative conditions like Parkinson's, Alzheimer's, and rare genetic disorders. And what we have done also is, for the first time, in the 100, we found that there are about 7 haplotypes, as I mentioned, the subcasts, subgroupings. And in the 7, we found one particular one was the most highly represented, the HV2A, and we used that to create the first Zoroastrian-Parsi mitochondrial reference standard. This is if you were to compare, so when most people do their sequencing, they compare it with the Cambridge reference standard, which is for the Caucasian population. And today that is the only reference standard known to be utilized. And today what we have done is we have contributed to global public data by releasing the first Zoroastrian Parsi mitochondrial genome. And then we also did a consensus genome, because as I mentioned, there were 7 haplotypes, and those 7 haplotypes were further broke up into 24 subhaplotypes, 25 subhaplotypes. So we created a consensus, meaning that all the SNPs got recorded. All the variants got recorded in the consensus genome, while there was one. So the reference genome can only be one genome, one person's genome. So that became the reference standard, and the consensus genome was a contribution of the 100 genomes. And the consensus genome, this is version 1.0. So we will be adding more into it as we do further sequencing and further analysis. So what we have seen is definitely that the Parsi community, all the haplotypes showed largely Persian origin of this community. And so we have quite clearly seen that. And there have been, of course, some groupings in the 3 of the 100 which showed certain inputs from the Indian population, sub-tribes of India. So that is what we have found, and the 100. And I think we are going forward ahead now with further analysis and isolating these variants to start developing them into biomarkers for predictive analysis for certain diseases. I hope I've covered all those questions.
Oh, you have covered the whole book, Villoo.
No, yet to come, yet to come.
So
Yeah.
actually, for the benefit of our listeners and viewers, I would say that we Parsis are believers of, we worship fire, and we do not use tobacco because we do not smoke because of our worship for fire, and therefore tobacco use is taboo in our community. And I can understand your interest in doing this work for trying to elucidate these Zoroastrian-Parsi genes. Now, and the diseases that are caused by tobacco use, such as cancers of the head and neck, the foot pipe, the mouth, and so on. So, what does your paper suggest? Does it suggest that these Zoroastrian-Parsi genes, which may help scientists discover biomarkers which are predictive of diseases which are caused by tobacco use, do you think that that can be helpful? Do you have some thoughts on this? I know that you have talked about it in your papers. Can you just elaborate?
Yeah, you see, while I had created the bioarchive, we had collected the samples, but the trigger for the sequencing really came when I met Dr. Derek Yak, who is the president of the Foundation for Smoke-Free World. And I proposed that, there's this endogamous community collection, which is a non-smoking collection. And why not we create the standard for a non-smoking pool and then link it to a larger study of smokers, non-smokers, and to arrive at an early prediction algorithm, a signature for creating a predictive diagnostic. And so I wrote up the grant, and we've been funded by the Foundation for the Smoke-Free World, which in fact is the reason why we could start this large study, because mind you, this is a very expensive piece of work to get done. And so so what we found was very interesting, that we found that most of the genes that have been linked to the smokers and certain signatures linked to smokers and carcinogenic susceptibility were not found in our population. And I know that most of the public may not understand, but mitochondrial genes, variants in mitochondrial genes like ND5, ND6, and transfer RNAs have been known to be associated with lung cancer and non-small cell lung cancer in other non-Parsi populations. And we did not find these signatures in this 100, which we had carefully picked and selected to be non-smoking. So we did not find that. And also certain mutations, which are known like G2A, etc., were found in less than 6% in our cohort. So it clearly indicates that this population, because of cultural practices and religious practices, have not smoked and therefore have less susceptibility to lung cancer and other smoking induced cancers. So, and I think this is going to be a big major part of the study, because we are now collecting samples for early stage cancer, lung cancer, and not among the Parsis, but in the general public, and smokers and non-smokers. And we will ultimately, from this large big data set, using artificial intelligence and machine learning, will arrive at predicting algorithm, which will then predict the outcome early on for patients and public at large. And we are using a specific methodology to look at that. Besides the sequence, we are looking at free circulating DNA and free circulating RNA. It's a whole technology there, which will, I don't want to, make it too heavy, this conversation, but ultimately by the year 2, end of year 2 and early and 3, by the third year, we will have a diagnostic which can predict presence or absence of a potential.
Bikaji is able to log in or not?
So yes, I think what you have done is that you very carefully studied the gene expression in your non-smoking Parsi population. Do you feel that this kind of a study can be useful or can be implemented to tailor adjuvant therapy for some common cancers, or like the cancers that we see in prostate, lung, the colon particularly, or breast cancer in our female patients? How much of your study will pave the path for this kind of personalized medicine? Because ultimately you have to give personalized treatment for different cancers. It cannot be one treatment for all cancers. And your gene expression study
Absolutely.
Yes.
Yeah, absolutely. I think our focus is on cancers, neurodegenerative diseases, and rare diseases. But just to tell you, this particular project, say, is a maybe 3-year to come to an algorithm. So, but in parallel, we are starting with other head and neck and esophageal cancer using the same methodology. Although the Parsi 200, we plan to finish 200 as a basis of the non-smoking population. And then we move on to the other populations and other diseases where we collect specifically for those conditions and arrive at these predictive diagnostics. And of course, precision medicine, because once these biomarkers are clearly identified and well known, and in parallel, we will start drug development. And I think we have the capability, and we have done it in the past. We have done, we have built 8 biosimilars in the farm. We are very well, shall I say, positioned to take on drug discovery. Of course, you need funding and you need right scientists. But I think this is such an interesting and important project. We will be able to get sufficient manpower, provided we are able to have the right pool of funding. We should be able to release precision personalized medicine in the future. And this is where we are going.
So you talked about increased association of mitochondrial variants, and therefore, of course, they would contribute towards an early diagnosis and treatment. Do you think these are the indicators to raise caution in our community? How would you be able to use this information for early diagnosis? Ultimately, we are interested in seeing that less people would die. Our population is declining, and we want to give your kind of research for early diagnosis, early treatment, and save our people from succumbing to these diseases. Do you think there would be indicators to raise caution? Would you have them?
Sure. I think it's very clear that we are facing a very tough time at the moment in our community in terms of numbers, and the dwindling population is a cause for concern. And however, we've shown that the increased association of variants with conditions like Parkinson's and Alzheimer's, I think we can address that to prevent the onset of early Parkinson's and Alzheimer's, because longevity is a trait we have, perhaps because we've inherited good genes from our ancestors, good quality genes. And so we live long, but the environment and the surroundings and the early onset of Parkinson's that we are seeing, as well as Alzheimer's, we need to find a an early detection system and a preventive measure also coming in to address the requirements of the community. So I think there's a big role to play that. Also, as I mentioned, we are seeing a lot of colon, ovarian cancers, and even just in the mitochondrial genome. Let's see what comes out of the nuclear genome. But we are seeing ovarian cancers, rare diseases resulting in inherited, I mean, almost many of the 100 had the lawn disease variant, which is visual disability, hearing disability, muscular dystrophy like diseases, and also infertility. So we have, with the isolation of these variants, we have to now look into the, what I would call mechanisms, the molecular and cell physiology of these diseases, and go deeper into it and see how we can understand what's going on and try to put a, prevention and precision opportunity in place. So, it's a very large project now, and each of these, addressing each of these conditions itself is a significant project. So we are working at multiple levels from grants to, writing specific academic partnerships and corporate partnerships as a way of accelerating this work, because you've already got this data with you. Now the next step is to move it next, to the next level.
So, Villoo, as I spoke earlier, we Parsis are non-smokers, basically.
Yeah.
And it will, of course, offer opportunities for scientific study as to how non-smoking can protect us against lung cancer and cancer of the mouth and food pipe and so on. And I know that the Smoke Free World Foundation is collaborating with you with the goal of making the world smoke-free. Global smoking cessation is their goal.
Yep.
And you are collaborating with them. What do you think? What do you think is their? What is the likelihood of this? Your research helping out in this?
I think it's going to be a major contributor, in my opinion. And it is very clear that this data, the genome, I mean, at the end of 5 years, we hope to have at least 5 cancers, biomarkers for 5 cancers, a diagnostic chip, maybe you may say, for 5 cancers in place, which can give a human being a predictive opportunity to predict that he is prone, or he or she is prone for cancer or not, as well as we are addressing Parkinson and Alzheimer's and its accompanying dementia, the whole triangle there. So we hope that we will be able to contribute and deliver the prediction chip, I mean, for all these 5 cancers and the neurodegenerative section of our work. And we hope that the foundation will support us to make sure that this is executed. I think there are very good chances of us being able to do it with the data we have at hand and with the new data pouring in.
So you spoke about this cancer biomarker development right now. And of course, they require to be validated.
Yes.
And then we have to have this cross-disciplinary integration. So how do you think, what do you predict is going to happen in the next 5 years in this field of cancer biomarker development? Do you think there is a need for this kind of a cross-disciplinary integration of science and clinical expertise for research?
That's absolutely the need of the hour. You're so right that in the next 5 years, we will have to link population genetics and to cancers, and it has to be individualized with specific groups for treatment, because the population genetics will be grouped, people will be grouped, and they will have their precision medicine post-diagnosis and specific targeted drugs linked to this subset of biomarkers found in each individual patient, leading to precision individualized therapies would be the order of the day. And I think and we will have to link with the clinicians for that. And so cross-disciplinary research, especially in cancer, is increasingly relevant and important to reducing the gap what is identified as best practice and what happens in clinical care. So the assimilation of diverse perspectives, research approaches, and types of knowledge is important in helping research teams tackle real-world patient care issues, create more practice-based evidence, and translate the results to clinical and community care settings. And that is where we are heading. I mean, I have to move towards that direction, and we are moving in that direction. And of course, we need, of course, the circumstances and the environment to play kosher with us and pull us along.
So yes, you have been leading this Avesthagenome project with great enthusiasm and great zeal. For what do you think this project will have the impact on our Parsi community? How will your research ultimately translate in helping the community? That's very important. I mean, the research has to come from the laboratory to the bedside, as we say, or to the patient, or to the clinical environment. How do you think your research is going to help in practical use for our clinical practice?
I think it's, first of all, I will say, Parsis have been extremely, philanthropic in their outlook. I mean, as you've seen over the years, we've built institutions, we've created
Yes.
all kinds of major industry, and we have contributed to communities in India and in the world. So in a way, I feel that this, the collection, the Avesthagenome Project collection is an endowment that will go on for years, and Parsis will be contributing to science globally. And that would be a major impact of philanthropy. And of course, for the community, the outcomes will be informed to them. I mean, what we are giving out, as soon as the sequences are done and analyzed, each individual, whoever contributed their blood to the system, will be given their data and will be advised on the next steps if there is any cause of discomfort. And also, while we have, all the outcomes will be partnered and perhaps monetized, the funds from this work will go back into research. This, it will, see, India absolutely needs a top quality systems biology institution. So Avesta Genome Project could be that institution which would also generate business. It will generate alliance fees, licensing fees, but all that would plow back into the running of this entity and would be a self-sustaining research model, and should be very interesting to see pull that off. And so to the community, they get their data, they get their, and any drugs or and any biomarker diagnostics coming out of this will be made freely available to the community. We will just provide it free. And in case we do alliances with different parties, third parties, it will be part of the clause in the agreements that the Parsi community will be given totally free treatment in these, whatever outcomes come from this work.
You know, Parsis have always promoted women education-wise, their education, then also given them empowerment. And actually the success or the level of civilization of any society depends on the empowerment it gives to its women. And again, now we want to shift gears to see, because you talked about the mitochondrium and the maternal mitochondrium. So how does this information from your study impact the women in our community? And women, and what about their women's rights? Something which I touched in my article very briefly today morning. What do you think about this? What do you think is the impact on our women and the community and their rights?
I think it's significant, the impact of this work. It clearly shows that the women are carrying the energy genetics, and they are pretty much linked all the way to Persia. So if we are, I mean, I don't want to go into that direction too much, but the women are inheritors of the old ancient mitochondrion, the Indian, the Parsi women in India. And yeah, I think they deserve to be honored, and they're given the equal rights that the men get in every which way. And we have to sit across the table with our community elders and work it out on the basis of science, because science is telling you this is the answer, you know. And so let science speak and let man-made laws be addressed through science. You know, let's communicate. I mean, we have to communicate and come to an arrangement that there's one law for all. Thank you.
I realize that I just triggered the woman in you. So tell me, how does this information from all your study now, you would like to give our community members some tips on how to manage? For example, you talked about cancers, you talked about Parkinson's disease in great detail. There are questions which families ask. What about the history of cancer in our family? Who should they go to? How should they try? What testing should they be done? How soon? And what kind of genetic testing should be done? And will that be helpful? Will gene therapy be helpful in Parkinson's? There are many questions which people have in their mind. I have been asked these questions so I thought, why not ask you to address them?
Sure. I think there are many new therapies coming out, and the world will go towards genomic medicine. I mean, clearly, some therapies are already out, you know, in the public. And as recently I read that, you know, that you can actually do gene therapy on the mitochondrion itself. And this was published 2 weeks ago. So, which are considered difficult to do, but it's been done 2 weeks ago. So I think the world is moving fast into, you know, what we call cell engineering or CRISPR-Cas based, editing based, or advanced cell therapies where they put back the gene or even replace the entire mitochondrion, which was also done. And I believe in Mexico, and the person is clinically stable today, you know, who got his mitochondrion changed. So I think mitochondrial replacement is also another one. This was a Leigh syndrome, transmission of Leigh syndrome, a form of a mitochondrial disease where the child, the mitochondrion was replaced, and the child is doing very well with a very low level of the mutant mitochondrial DNA present in his bloodstream. So I think it's possible. Technologies have really come into place. They're very solid. And it's about us getting, isolating, identifying, deep diving, and understanding the biology and physiology and designing around it with the toolkit that's come into place today, like CAR-T and CRISPR and many others as advanced cell therapies that have now come.
It's good for the neurological problems that our community suffers. You know, for many years we knew that there was no scope for neuronal regeneration. For example, nerve cells don't regenerate. But now we know that there's definitely the complete shift in the thinking. And again, your mitochondrial replacement therapy may be of extremely great use in the regeneration of neurons too. And this may be helpful in the diseases of, let's say, Parkinson's disease, for example.
Yeah, sure.
Now,
I think, I mean, as I said, the toolkit is there.
yes.
We need to put it together.
Yes. I know that your project is now very, very vast, as you already hinted. It is a very costly project. You need funding for this. And I heard you talk about something like $100 million are required. What do you think? From where will you get all these funds? Are you ready for that? It's a big deal.
Yeah, I've been on the road, shall I say, for the 20 years of my life, from 2000 onwards, when I started actually stepping out into the big bad world from academia. So I've been pretty much a good, reasonably decent fundraiser, you know, and have built out a lot of streams of work. Of course, you know, there are events that take place around you, like the 2008 crash and now the COVID, that are out of your control. But you you have to ride these hurdles and overcome them and keep moving on. And I believe that it's possible to raise 100 million for this kind of a project.
We have many good.
I think if I'm given a little bit of support for overriding the next next couple of shall I say quarters, I will be able to march into the world and collect that money for this project.
You can depend on some of the very prominent Parsi businessmen who have got, very philanthropic. Charity begins at home, we always say that. So let's hope they loosen their purse strings and help you in your project.
Oh yeah, they've been there. I know they've been good people, and I've had fundings in the past from the Tata Industries and Godrej Industries, and they've been fantastic people. And yes, we will go. We have just about got this project moving now. Some more publications, some more work, and I'll be out there again.
You just talked about, very briefly, you just touched, and I thought I should interrupt you for that. You just talked about the coronavirus. Now, this is something which is now keeping us awake and alert and afraid also. If there's time, if Meher allows us the time, then we can talk a little bit on coronavirus. How do you think? Is it okay,
Yes,
Meher, if we just
go.
talk a couple of minutes on
yes, please go ahead.
Because Villoo has expertise on all these extraordinary, difficult kind of problems. Let us see what her views are on the coronavirus, and do you think this virus is going to stay for some more time? And I know the answer is true, but let me see what you think about it.
You are the doctor. You got to tell us what's happening in the hospitals.
Yes. Oh, yeah. I should put some pictures of my PPE suits and so showing you how we are operating. Yes, but work doesn't stop. But it's, of course, very much low key.
Yeah.
It's not turning out. And we suddenly wonder what happened to all these people who used to throng our outpatients and the way we used to sit outside my office. Now we suddenly find them not suffering at all. Or they are too scared to come to hospitals, or they are just overawed or overwhelmed by this virus. What do you think about it? Do you think there's something about the vaccine? Now every day we in the newspapers, on one side we talk about morbidity and mortality. On the other side, we talk about some companies which are becoming ever energetic and talking about some vaccines. And I don't want them to rush to something hurriedly. What about human trials? Is there any out-of-the-box treatment for this COVID? And of course, the world has already changed a lot, as we can see. So I can only dread to wonder what's going to happen post-COVID in the next year, 2 years, 5 years, I don't know. What do you think about it?
Yeah, I think the world as we knew it prior to COVID-19 no longer exists. So since December 2019, the novel coronavirus has rapidly changed the way people work, play, socialize, relax, essentially, and the way they live their lives. So it's horribly unfortunate because it makes me very sad to see that era come to an end where we were, or how carefree we were. So I think we need to see the virus is also has many strains, and so the vaccine and the human interaction. Now recently we heard that they have found 3 biomarkers linked to in the human being that could perhaps tell you whether the drugs are working or not, or you're susceptible or not. So it's a human viral interaction. There are many viral strains. There are many types of humans. So the vaccine that comes will have to be all encompassing if it's one for all vaccine, or you may need different subsets of vaccines, even like in the treatments. And I'm sure you know it, that there are all these mixtures of drugs people are using, hit and miss combinations. Some survive, some don't survive. So we have to understand the viruses completely, as well as the human genomics, human genetics, and see what's going on there. And because these viruses are RNA viruses, they can change. and they belong to a family of viruses that cause illnesses from common cold to severe acute respiratory syndrome. And so the risk of dying from COVID-19 can vary considerably depending on age, ethnicity, access to healthcare, socioeconomic status, and underlying health conditions like heart and lung disease or diabetes. So I think as of the current situation, the world seems to be headed in a long-winded battle with the virus. There are a lot of factors that would undermine and determine whether the earth is free from it. And it is unfortunately tied down to many uncertainties about this virus and the cause of the disease. So we need more information. I think the information has to be coming in, and we need to do more further deep research on these viruses. And of course, there are two parts. One is the vaccine immunization, and the second part is herd immunity. And in the recent study in Science, it was shown that 43% to achieve herd immunity for COVID-19, and it's difficult to do. And vaccines, there are many in the pipeline, as you all know, and there's the US-based Moderna, Oxford University / AstraZeneca, and the Russian coronavirus was recently announced, and of course the ICMR Bharat Biotech from India, and of course the Chinese ones. So, but I agree with you that without a proper clinical trial, we should not be, of course we should encourage vaccines and they should come out, but we need a proper clinical trial for it. And out of the box thinking, there are many things we can do, and people are repurposing certain anti-TNF alpha drugs, and I think that is another approach for enhancing immunity, and also screening and repurposing existing botanical bioactives specific to specific viral proteins, and which could provide a rapid and iterative identification of molecules to validate different viral-based how do you call it, nutrition-based, nutraceutical-based outcomes.
Yeah, I think, yes. Meher, I guess I'm just very sorry that we have overstepped our time.
It's all right. It's all right. Carry on. Please carry on.
So I think Villoo has done an enormous amount of laborious research in our labs. And I think the viewers will realize the importance of this kind of work, basic lab research, in understanding the various connotations of so many diseases, especially our Parsi heritage, which is so important. And we will come to some solutions with your research work in understanding our genes and in seeing how we can, our endogamy on one hand has given us some advantages, but on the other hand, it's also given us some disadvantages. So we need to, like old age itself, sometimes carries so much of a problem of osteoporosis, osteoarthritis, and fractures, and dementias, and strokes, and whatnot. So we hope that your research will enable us to go deeper into our ancestry, into our heritage. I know that we have a very glorious past, and we see a pretty vibrant present, and we hope to have something useful from your lab research to give us a promising future, and to see that our numbers, for some reason, our numbers should increase, and not only quantity, also quality-wise. We don't need quantity, but we should increase our numbers because we already have that quality.
I agree with you, totally there.
Yes. So thank you very much for sharing your great insights and your great lab work. I know we can't do justice to that in a matter of 45 minutes or an hour. And we hope we can read some of your papers and write-ups in future, and also see you on a bigger stage to help our community move forward. Yes, thank you so much.
Thank you so much, Keki. Thank you. It was a pleasure getting to know you over this whole dialogue, and look forward to working with your hospitals and gain your knowledge. I think the clinicians' knowledge, I mean, you guys are out there with the with our patients. And I look forward to engaging more with all of you.
Thank you.
Thank you for leading this conversation.
Thank you.
Can we take a few questions, please?
Sure, I'm fine.
Yeah, one question is, you mentioned non-smoking section. This is lifestyle related. How is it related to inherited genes?
So smoking in inherit, that's what we are saying, that in the non-smoking population, we have not seen, there are changes in our genes, from say a decade to decade. We are just looking at the epigenetics profile of DNA collected 10 years ago and today. And we see a lot of what is known as methylation of certain CPG islands, they are called. And so when we say that the Parsi population hasn't smoked for centuries because of social practices and respect for the fire, it means that over the years, there are certain susceptible loci in the DNA. They have escaped that, while the others who have smoked have been mutated there. So there's no inheritance involved in smoking. It is involved, but by habit and social. That's why you've got a non-smoking population who has less of those potential cancer-causing mutations compared to those who have smoked.
What is the title and who is the publisher of your work?
Who's the publisher? Okay, so the title of the paper is the first Zoroastrian-Parsi reference mitochondrial genome and genetic signatures and their implications. That is it. And it was published in BioRx4. It's an open journal where you go and publish it. And after it's like a preprint, you release your data there. So it gets its date and position, and there's no, you establish your first in the world position. And after that, it goes into a publication in a journal, peer-reviewed journal. And at the moment, we have sent it off for to the peer-reviewed journal at the moment. It got published in the BioRx.
Are you partnering or considering partnering with other global efforts looking at maternal haplogroups or mitochondrial DNA?
Of course, we are, definitely. We reach out to a number of groups. We work with a number of groups. And we are constantly actually Avesthagen's model is partnering. We generate early research, and then we find partners globally, academic and and industry, and take those early discoveries to the next step and to the next step, etc. So that is what we do.
With the possibility of mitochondrial replacement, could that be used proactively to treat Parkinson's and Alzheimer's?
I think it can be used. It's a little early stage, and we have to see a few more cases. But yeah, it's possible. The technology is possible, potentially possible.
It has to be legalized, isn't it, Villoo?
Yes, of course, it has to be legalized. I mean, that's besides the point.
Legal questions.
UK has not yet. I mean, they have not yet legalized it. Yeah.
Have you done any research on genes connected with G6PD or anything that indicates the frequency of celiac disease in Parsis?
We will analyze it because now the data is with us. So we have finished the whole genome sequencing as well as the mitochondrial sequencing. We have all that raw data. And so you have to basically work on that data. So we can look at we can look at the celiac disease. We can look at G6PD. We can look at, now this project that we have been granted funding for is aimed at lung cancer. So, but that doesn't mean that the data that's being generated cannot be quizzed for the others, because the answer is there. Of course, you have to build the whole team around it, and you have to, mine it and then convert it. So yeah, it's all possible. We have the data with us. That's the beauty.
Okay, this is a lighter one.
Hmm.
Why do we see girls more intelligent as compared to boys in our community?
let's ask that to Dr. Turel. Professor Dr. Turel.
Because of women empowerment. we men have always have have a soft corner for the ladies and give them more power. You've seen so many homes are matriarchal in the Parsi community.
True. Yeah, I've seen a lot of intelligent men too, but I think I mean, I have my theory that in the 60s and 70s, things got too comfortable. And they had a house, they had so the boys became too comfortable and they didn't want to leave their homes, and it was just their little world, and they didn't want to adventure, while the girl had to fend for herself in multiple ways. So she went out and did things.
Like you did.
I grew up in a very, very, free-minded household. So we all worked, boys and girls.
Yeah.
I think that ends the question answer session. Adel, would you like to take over?
Hi, Adel. May I invite you to give a vote of thanks? Adel is, of course, the global president of WZCC. Adel, we invite you to say a few words.
Yeah, thank you. Today's talk, Our Genes, Our Future, Unraveling the Ancient Zoroastrian-Parsi Genome, was indeed very interesting, timely, and useful. Thank you, Villoo, for the very interesting and stimulating talk and the well-answered questions. Thank you, Keki, for the very pertinent and probing questions you asked Villoo. Thank you, the audience, for the record participation of 292, and for your questions which demonstrated your interest and enthusiasm. Villoo, with your help, we now understand better where we came from and how our ancient genes have impact on who we are, how we behave, our idiosyncrasies, the genius within us, as well as the physical and mental issues we are destined to be challenged with. The talk helped us to understand how religious taboos have protected us from certain cancers and diseases. The paradox of longevity, but with prevalence of breast cancer, Parkinson's, and dementia. Also very interesting is the fact that our genes are dominantly from the mother's side. How Avesthagen is contributing in the field of science and innovation. The study will enable us to proactively work to protect us, or at least slow down the negative impacts. We cannot afford to gamble and must take care of ourselves and the community with tender, loving care. Villoo and Keki, in appreciation, WZCC would like to mail you our logo pin, which we hope you will wear with pride. Finally, a heads up that we are planning many such important webinars in the future, and stay tuned for details. Thanks everyone for attending. Stay safe, stay healthy. Thank you.
Can I say one? I would like to thank the community, because without them, I would never have been able to reach where I have. And in this elucidation and analysis, without their contribution, they came to all those blood collections and were fantastic participants, and I owe them a lot. And I'm deeply grateful to the community, the Zoroastrian-Parsi community of India. And also, thank you WZCC for helping me put this, for inviting me to this podcast, webinar. And I really appreciate that. And just a few things I want to mention is that why this application of this science from our community is for the global public too. It benefits our community as well as the rest. And because it's just that we are using ourselves as a prototype. I mean, we are using the outcome from our genome is useful to the world at large. And mitochondria is one part of the journey. There's the whole nucleus still where the data is just out and we will be analyzing it. So it's going to be, you will see much more coming from us. This is not this, it's only the start. And it's applicable to men and women, community, our community, and to the world. Thank you so much, everyone.
Thank you.
Right.
Thank you, Dr. Turel. Thank you, Dr. Villoo Morawala-Patell. Everybody has appreciated your session, and we look forward to you helping the community in many more ways. This was the whole focus why we had this session. Thanks a lot, everyone, the participants. We were very happy, or we are very happy with the questions that were sent to us. And you will be seeing the video, the recording of this session on YouTube after 24 hours. We'll send the link, we'll circulate the link. Thanks a lot to everyone for participating. Thank you.
Thank you.
Thank you.
Thank you very much. Thank you.
Thank you, Keki. Thank you, everyone. Meher, thanks a ton. And Yazdi, and
Thank you.
all of you.
Bye.
Thank you.